Okay, so I'll be showing some of preliminary findings in our attempt to sort of map the evolution and trends in the field of metabolomics with some natural language processing techniques, and um, but to really start with some premise. So about over 1 million of biomedical and life sciences papers are published every year. So comprehensive mastery of scientific knowledge is just impractical. Even at that, keeping up with papers that's published even in subfields can be very challenging. So that means we need a set of tools and techniques and methods that allow us to sort of pass through information from a global perspective, rather than tracking down individual papers. And we have an hypothesis about this, and is that unsupervised word embeddings will capture latent knowledge from the literature, and in this case, metabolomics literature, that will allow us to really answer questions from these global perspectives. And for our data sets, we use the publication abstracts containing the words metabolomics, metabonomics, ranging from about 1998, when the field actually emerges, to January, February 2024. And we have about 82,000 entries for these data sets. For what the data set looks like, you can see here you have the Type 2, you have abstract, journal Type 2, publication year, authors, and so on. And then we also added another column um, that actually label each of these rows, so each of these data points, by inferring the title or the label using the journal title. So to give you an example here, this is a paper published in Clinical Kidney Journal, so then that category will be nephrology. This is one on neuro-oncology advances, so this will be on oncology and things like that. With regards to generating embeddings, we used an encoder. So we use an encoder. So for those who are not familiar, an encoder is a type of a transformer model um, that essentially allows you to understand the sort of semantic relationship between words. This is very different from, say, decoder model. And if you've used ChatGPT, you've used that kind of model, because ChatGPT, for example, is a decoder model that basically predicts the next token. In the case of an encoder model, the way it's pre-trained is you have this, for example, I am, then you max this word happy. And then the goal of the encoder model is to really figure out what is in the maxed section. And by doing that for a large corpus of words, so for a large corpus of literature, the model actually learns a semantic relationship between words. So that's basically the big idea here. And case in point, we use the PubMed BERT. So this is a domain-specific language model exclusively trained on PubMed datasets to generate our embeddings. So basically, this is not very clear yet, but basically what's going on here is the model takes in words, in this case an abstract, and it gives us out a 1 by 768 embedding. So before we start looking at embedding figures, let's just look at some publication analytics. And basically, we see these trends of publication, metabolism publication by year increasing. And here I was able to sort of isolate what journals publish the most metabolomics papers. And clearly you can see scientific reports followed by metabolites, followed by plus one, and so on. And I also attempted to identify which authors are the most published in the field. But the first time I was presenting this in lab meeting, a Chinese postdoc said what you have successfully done is actually to plot the most popular names in China. So this is most likely wrong, and now I'll be attempting to sort of pass this through some other methods. We also have a word clouds of abstracts. So these are the top 100 words that are present in the papers. So the most frequent words: LC, biomarker, liquid chromatography, and a little p-value. So those are the tiny ones perhaps that you might not see. Distribution of abstract lengths. The modal word length is about 200 words. This is basically plotting numbers of characters on the left side and this side, the number of words. on these other sites, and then we have these sort of publication number of papers. Spikes actually corresponds to journal titles. We think it corresponds to journal titles, so you can see the spikes right over there. So basically, what's going on here is that we do have these 1 by 768 embeddings, but we can't visualize that. So to visualize that, we're going to use a dimensional reduction technique, in this case t-SNE, that helps us reduce a 1 by 768 vector to a 1 by 2 vector. And once we have that vector, then we can plot it in the graph that we all can see. And this is labeled by that inference from the journal title that I talked about. So the next thing I'll do is I'll just kind of like walk you through this map, and let's look at some sort of obvious clusters. Plant sciences, environmental sciences, animal sciences, pharmacology. We can see cancer biology there, and then we can also see immunology. This is not shown, but this is immunology, and essentially it's hanging out with cancer biology, and of course that shouldn't be surprising. We have cancer biology, and we have analytical chemistry. So let's also attempt to peel out and just kind of like focus on some categories in these embeddings. Before we do that, we see these sort of combinations of colors, and then there's so many things going on. We can't see what's going on very well here. But one first sort of idea will be this is perhaps an interdisciplinary space, but more on that later. Plant biology is the most isolated clustered field, so we're basically focusing on plant biology now, focusing on that, and that's quite interesting, just observing this visually. Analytical chemistry is all over the map. Okay, so analytical chemistry is all over the map, but and that's of course not surprising, because of course metabolomics depends on analytical chemistry techniques, and there's this focus of analytical chemistry at the on of the map. Okay, even though it's dispersed throughout. We also have overlapping fields. So this is toxicology. I'm drawing just these black circles over that. And by including environmental sciences, we can see the overlapping between toxicology and environmental sciences. And we have other instances of these, such as cancer research and immunology, animal science, and food sciences. Another interesting thing we can do with these embeddings is that we can do keyword highlight. So I can basically go through all of the abstract and basically write a program to just identify which abstract actually mention a particular phrase. So for example, I'm saying what abstract mentions the term for the first time, and I want that to light up with these black dots on my graph. And then basically that could denote some sort of novel finding. At least that's when people would use that term. I did that with COVID-19, and then basically we get this really nice cluster that will be observed or will be investigated further. So many things comes in handy with this. We can check the presence of p-values, and the p-values abstract containing p-values actually have these blue spots that have been indicated. And an interesting thing is that for some reason some sections of the map just don't mention their p-values in abstracts. Why is that? Well, I suppose these are some of the interesting questions about trends. And we can look at abstract lengths at some of the plots we have, title lengths, co-authors count, reported sample size, just basically visualizing that with these embeddings. So going forward, one thing that we'll do to basically understand trends in fields of research is basically to have these visualizations of research clusters to debate on keyword presence and publication trends. And how do we do that? We would isolate a particular research cluster. Then after isolating a particular research cluster, we will do keyword-based labeling, so basically using some specific keywords or key phrases and titles or abstract of papers. We use that to label those data points, those abstracts, and then we can basically visualize these research clusters based on publication year, based on keywords, and then see is there any trends that we can actually find. And for the elephant in the room, something that I didn't mention is the fact that up until now, even though this is part of our embedding map, it's not obvious by looking at the embedding maps unless you zoom in. But the reality is that the vast majority of these is actually unlabeled. Okay, so this is about half of the data set that we have, and that's just because we use this method of inferring the label from the journal title. So papers published in journals like Science, Nature, PNAS are just not classified. So going forward, the main next step that I'll be doing is basically to this is AI driven approach of actually labeling the papers. And this could be something like clustering these embeddings using a clustering algorithm, say HDB scan, k-mean, say, take those abstract in the clusters and then pass that through a large language models, for example, an open source language model like Llama 3, or maybe a more appropriate one for scientific corpus. I don't know yet. But basically, that's basically what I'll be doing is to do topic modeling to basically allow us to label all of these abstracts. Okay. So that's what I have. Thank you so much for watching this poster talk.